is going to take us through the cell. So hopefully we are now past chemistry. So if you're one of those people like I am not a chemistry person, I do not want to do chemistry again. We will not be doing chemistry again. We will be applying it, so there's going to be times where you might have to tell me what an amino acid is by seeing that it has an amine group on one side and a carboxyl on the other. But you aren't going to be, we're not going to be talking about chemistry in particular. We're going to move on to what people usually think of when they think of biology, things like cells and things like the organelles. Remember organelles? Yep. Nope. Give me an organelle. Anybody? Mitochondria. Everybody loves their mitochondria. Powerhouse of the cell. What was that next? Chloroplast. Powerhouse of the plant. Yeah, cool. I got two. Anything else? Cytoplasm. Ribosomes. Good. There's a big one that I haven't heard yet. Nucleus. Yeah, the nucleus. Cool. So that's what we're going to be doing when we talk about that, when we talk about the cell. But let's do a little background before we get there. All living things are made of cells, or at least one cell. If you do not have at least one cell, you're not considered alive. So one of the things that's not considered alive, but still very damaging to us, is this little guy called a virus. A virus is not considered alive because it doesn't have all the components that you need to be considered living, and we'll talk about that. Go on and get to something that we actually need to talk about, which is okay. So cells are usually something that we cannot see without the naked eye, or without a microscope. We can't see it with a naked eye. So in order to see it, we need to visualize it with something like a microscope. Light microscopes you've probably worked with in your freshman, sophomore year, probably worked with them in middle school. And we're going to go through some of these pretty quickly. But here's just kind of a scale of it. Let's see, here's a better one. Human being has many, many cells, and we can obviously see a human being. Here's one that you can see that is a cell, and that is an egg, chicken egg. A chicken egg is a single cell. Unless it's fertilized, then it's actually 2 cells and 4 cells, etc. For some of the more detailed work, if you want to look at things like bacteria. Oh, that's not what I wanted. Bacteria. These guys here. Not that one. That's not a bacteria. That's a mitochondria. This guy here, bacteria. You need a light microscope. If you want to see something that's really small, you're going to use something called an electron microscope. And that will let you visualize even smaller bacteria. It'll let you see things like organelles. So that mitochondria that you talked about will let you, the electron microscope. Electron microscopes are really expensive. They're really huge. We don't have one here at the school because it's millions of dollars to have one of them. And they're the size of a room. They're big. What we are going to really be focusing on is these things called organelles. And organelles are structures within the cell that have a membrane around them. You got one. I gave you one.
Yeah. Oh, you
I gave you one.
Oh,
Is that yours? Yeah, okay. So organelles have a little membrane around them. They are found in a certain type of cell. And I just want to see if I can find that. I'm going to skip over. I thought I had edited these to take some of this stuff out, but evidently I didn't. So light microscopes can see things that are up to 1,000 times the actual size of the specimen. If we want anything more detailed than that, we need an electron microscope, electron microscopy. That's kind of hard to do. Here's a light microscope. There's a bunch of different cells that we have here. Here's a light microscope as well, but these have been stained in different colors. And then this last one down here at the bottom is actually an EM microscope or an electron microscope. And these are little cilia that you see here coming off of this cell. All right. Not worried that you understand or remember the differences between these microscopes, so I'm going to actually just do that. Starting early. Same thing with this one. Could have sworn I already did this, but I guess I didn't. A very typical type of procedure to help visualize different structures is called cell fractionation. And what that does is it will separate parts of the cell from each other. And that can be helpful in trying to determine the structure of a particular organelle as well as the function of that organelle. You use a centrifuge, and when you use a centrifuge, anyone used a centrifuge before? Anyone know what a centrifuge is? I should start with that question. Yeah, so it's a spinny thing, right? So the point is you can put in these cells, and the parts of the cell that are heavier, as you spin it around, you have it in a test tube, and you spin it around in this machine, and as it spins around in the machine, the heavier parts are going to sink to the bottom, and the things that are lighter are going to be on top. And so here they're showing you that there's a pellet on the bottom, and that is going to contain the things like the organelles. And then you can start to look at the organelles, whereas this light stuff up here, that's going to be things like the cytoplasm that are liquid in content. And you can just keep doing that until you get the part of the cell that you really want to look at. But we're really more interested in the organelles themselves because we're not going to talk as much about how they get the information as much as we are going to talk about what that information is. So eukaryotic cells have, let me just check here and see if that, yeah. So there are two different, actually going to go down two slides down to this one. There's two types of cells. There are prokaryotic cells and there are eukaryotic cells. We're going to start off with the prokaryotic cells. Prokaryotic cells are the simpler of the two types of cells. Every cell in the world is either a prokaryotic cell or a eukaryotic cell. Prokaryotic cells do not have a nucleus. They do have an area that's called a nucleoid. And in that area, they will have their DNA. So their genetic information will be in that area. But there's no membrane around that nucleus. And that's what we call a nucleoid. There's also no organelles. There's no membranes around any structures. The only membrane that there is the plasma membrane. What's another word for the plasma membrane? It's also the cell membrane. It's the same thing. Prokaryotic cells do have cytoplasm. Cytoplasm is kind of a semi-liquid that makes up the majority of the cell. We'll talk about what's in the cytoplasm as we go on. Let me see. All basic cells are going to have a couple of different features or a few different features. They have a cell or a plasma membrane. They have this thing called a cytosol, which is also called cytoplasm. They all have some type of genetic information that's carried on a chromosome. They have at least one chromosome. And they all have, prokaryotes, eukaryotes, both have ribosomes. Ribosome's job is to make proteins. If you remember from last unit, we had RNA and we had DNA. DNA stayed in its location, and RNA moved the information, a copy of that information, to the ribosomes. So ribosome's job, once they get that RNA information, is to make a protein from the genetic information. Yeah, wrong button. There we go. Back to prokaryotes. These are a couple examples of prokaryotes. They are in general, a lot of prokaryotes are bacteria. And some of those bacteria are harmful and some of them are not. You have antibiotics. Antibiotics work on bacterial infections. Strep throat would be an example of a bacterial infection. So pretty simple little critters. Okay, that's prokaryotic cells. We've got ribosomes, we've got cytoplasm or a cytosol, we've got a nucleoid but not a nucleus. We've got a plasma membrane, and that's about it. That's about it. They have a chromosome, but they're not as complex as eukaryotes. Eukaryotes have their DNA in a nucleus. And we call it a nucleus because it has a membrane around it. It has a full-on membrane around it. They also have membrane-bound organelles. So again, a membrane around those organelles. They have a cytosol that we call cytoplasm. Again, just a semi-liquid area that makes up most of the cell. In general, eukaryotic cells are going to be bigger than prokaryotic cells. And so when I showed you that picture of the human being, and then there was the egg cell, and then I showed you bacteria cells and the mitochondria. If you looked at an egg cell comes from a what? Well, usually, sometimes, I guess, depending on what animal. If you get it in the grocery store, it's probably coming from a chicken. Yeah. And a chicken would be an example of a eukaryotic organism. Okay. So a chicken egg cell is much bigger than, say, the bacteria that causes strep throat. You can't see strep. You can see the symptoms of it, but you can't actually see it. Okay. The plasma membrane. We have a plasma membrane around the prokaryotic cells and the eukaryotic cells. And its job is to kind of guard the cell so that only certain things can get in. So we call it a selective barrier. And we're going to examine how it's selective and why it's selective. Oxygen usually can get in. Nutrients can get in. Water sometimes can get in. Waste can get out. But if there's a toxin in the environment, we don't want that toxin to cross the plasma membrane, because if it does, it could cause all sorts of issues. So we want to keep that from happening. That's why it's selective. Here's a picture of that cell membrane. A big part of the cell membrane is going to be this thing called a phospholipid. Remember phospholipids? Yep. I hope so. What was unique about a phospholipid? You remember? What she said? Yeah, good. So there's hydrophilic areas and there's hydrophobic areas on a phospholipid. So if this is water out here, then these little heads, these are the ones that are hydrophilic. Yeah. And then the ones in the middle, sandwiched in the middle, they're like, oh no, it's water, run away. They're hydrophobic. Okay. That makes up a majority of the cell membrane, those phospholipids. There are other things as well, but the majority is made up of those phospholipids. See, we're still talking about chemistry, but now we're applying it. Shh, we won't call it chemistry again. All right. When we talk about cells and cell size, how many different cells can you see with your naked eye? Well, I can think of a chicken egg, right? Or a quail egg or bird eggs, right? But in general, you cannot see them with your naked eye. And the reason for that has to do with the size of the cells. In general, for a cell to function properly, it has to be small. And that has to do with some mathematics. Pretty simple mathematics, but it has to do with a little bit of mathematics. When you're talking about how fast something can get into a cell, first of all, it's got to get through that cell membrane. So it's got to get through that phospholipid layer. And second of all, some things can go and some things can't get through. But how fast it gets through is going to be determined on a couple of different things. And that is, actually, let me just see if that's down here. No, it's here. Okay. And that's going to be how much surface area the cell has. So on the outside, how much surface area does it have? Compared to its volume. Okay, so how much is inside? What happens is as a cell gets bigger, its volume grows at a much faster rate than its surface area does. So cells that are bigger have a lot bigger volume in them. And because of that, it's harder to get, say, if you've got a nutrient that you need to get to every part of the cell, if you have a larger volume, it's going to take longer for that food, whatever it is, a nutrient, to get to every part of the cell. Okay. We are going to actually do a lab on, and well, we'll see. I mean, COVID's over, but it's not over, right? It's never going to be over again. The reason I say that is because there's a lab that I do with potatoes, and it's on how fast nutrients move into a cell. And at the end of the lab, we've made all these little potato cubes, but it just always makes me want French fries at the end. So, in years past, I have been known to French fry some potatoes in here, and then give them to people afterwards. We on board for French fries?
Yes. Yeah.
What's that?
Can we get like one?
Wouldn't have to. Wouldn't have to. Okay, so here's a diagram looking at the surface area of let's start over here, of 3 different cubes. One is, you could say it's 1 millimeter. It doesn't matter really what the unit is, but 1 unit. And then you have one that's 5 units next to it. Let's just look at those for right now. When you look at the surface area, you take the height times the width times the number of all the boxes, and you get, so if it's a 1 side or 1 unit, you're going to get 6, right? 6 sides on the cube, each one is 1, and you get 6. Volume is also going to be 1. Sorry, is not going to also be 1. It's going to be 1. So when I look at the surface area to the volume, in other words, if I look at 6 divided by 1, surface areas on top and volumes on the bottom, my number's 6. But if I look at my 5 millimeter or whatever unit cube, my surface area is 150 now, if it's 5. But my volume has gone up a lot more quickly. So now my number is 150 divided by 125, which gives me 1.2, a ratio of 1.2. So the 1 cube is 6, and the 5 cube is 1.2. In order for a cell to be efficient, in order for a cell to be efficient in terms of moving nutrients in, you want that number to be as big as possible. So which is the bigger number? 6. And that's the what cube? It's the smaller cube. Okay? Bigger number, smaller cube size. Okay. Bigger cube, smaller number. Okay. So that's why in general cells are really small. Now you can make up for this if you're a single cell critter. If you're a eukaryote, there are eukaryotes like Euglena and Paramecium. They're just 1 cell, and they're just floating around. Those guys are both water critters, so they're swimming around, and they're just 1 cell, but they're small, right? But if you're say you're a human being, you still want your cell size to be small. So instead of, we can't just be 1 big cell, that just wouldn't work at all. Instead, we divide up the body into lots and lots of little cubes. So the body becomes multicellular, and each one of those cells is really small. So if we try to see just one of our skin cells or one of our liver cells or heart cells or whatever, it's going to be small, something that we'll need a microscope to see. Okay. All righty. Really looking forward to that potato lab now. I'm not going to do organelles yet because I want to give you some time today.